Well, hello everybody. We continue our discussion on cells and cell organelles in eukaryotic cell. Right, so this is a little animation showing us the cytoplasm with the organelles, the nucleus, central nucleus. This is the typical animal cell. So the nucleus is central. You can see the sausage shape mitochondria floating around, and the Golgi body, and so on. Alright, so let's get into it. So essential questions we'll be asking are as follows: what are cell organelles? The structure and function of cell organelles. Then differences between plant and animal cell. So at the end of the class, we want to, be able to answer these questions and more. All right. So what's a cell organelle, or what are cell organelles? So generally, organelle means little organs, membrane-bound structure within a cell, found only inside eukaryotes. So we know that with the prokaryotes, they have some of the organelles, like ribosomes, even though they are smaller. Then we say that they do not have membrane-bound nucleus, and so on. So the organelles are limited in other cells apart from the eukaryotic cell. So a cell organelle is equivalent to the human's organs. Okay, so we want to start off by looking at the cell membrane. The cell membrane is not an organelle, but then it is important that we look at it. Okay, so we know that it is the boundary surrounding all cells. Okay, prokaryotic, plant cell, animal cell, and so on. Whether the plant cell has a cell wall or otherwise, there will be a cell membrane. Alright, so we can see the structure of the cell membrane in this animation, and as a boundary or a layer or some kind of thin wall surrounding the cell is going to permit some materials to go in and out of the cell. Okay, so let's look at the structure. We said that it's a phospholipid bilayer. Okay, so the cell membrane surrounds the cell, so it's the gatekeeper. If we go to the club, that would be the bouncer, allowing some people who are of appropriate age limit to go into the club, and those who are not will be sent out. So we have the cell membrane made up of two layers, as you have said. It's phospholipid bilayer. So the bilayer, bi means two, and then two layers. So the membrane itself is made up of two layers, made up of phospholipid molecules. That's one end of the layer. excuse me. Made up of a phosphate, and then the tail. Okay, so the head is the phosphate, then the tail is the lipid. Excuse me. Right, so you have here the phospholipid molecule, the hydrophilic head. So the head is the phosphate, and hydrophilic because that part of it, attracts water, or is water-liking. And then the tail, which is the lipid, as you already know, hates water. Mostly lipids, fats, and oils do not have the affinity for water. They do not mix. So the major function of the cell membrane is to allow materials in and out of the cell. Okay, so you can see it here, where some molecules are going through this protein. Alright, so apart from the phospholipid bilayer, we know that the membrane is also made up of proteins, and these proteins essentially allow certain material that cannot go through the bilayer. Usually those materials that are water-based, some ions and large molecules cannot go through the bilayer, because, as we've said, the tails are water hating. So because they are in water, the ions and molecules are in water, they cannot go through the bilayer, and therefore, importantly, we have the proteins that allow or transfer materials in and out. Okay, they are specific for particular materials that must go in and out. Okay, later on, when we are looking at nervous system and respiration, you get to know that there are some proteins that transport particular types of ions. So sometimes we have the sodium gates, the potassium gate, the calcium gate, so on. So those are proteins that transport particular types of ions in and out of the cell. So the proteins have two types. You have those that are close to the membrane, okay, the outer membrane or the inner membrane. Those ones are the peripheral, or they are on the outside, peripheral proteins. And those that cross the membrane, they are the integral proteins. As I've said, later on, based on their functions, you have different types of proteins performing different roles. Alright, so we move on to the cell wall. So this is the rigid barrier around plants, bacterial and fungal cells. So it's not only in plant cells or in plants that we have cell wall. Right, bacterial do have, the fungal have it too, and they are all made up of different materials. So usually in plants we say that it is made up of a cellulose cell wall, because the polysaccharide material cellulose forms a major part of it. And then the bacterial cell wall is made up of a material similar in insects, as the chitin, or some people say chitin. So we say chitinous cell wall. Okay, then the fungal is rather the chitin. Okay, so the bacteria is made up of the peptidoglycan. Right, the peptidoglycan, which is basically two materials, amino acid or protein material, and then carbohydrates or glucose material. So that forms the major part of the bacterial cell wall. Then the chitin is rather in the fungus. The fungus have the chitin, which is also a form of polysaccharide or carbohydrate, forming the cell wall. Okay, so it's generally outside of the cell membrane, outside of the cell membrane, if it is present. Of course, for animal cells that do not have a cell wall, then it is only the cell membrane, which is the outermost material. Okay, so in plant cells, made up of cellulose, which is fibrous material for protection and structure. Okay, then we look at the cytoplasm. Of course, the cytoplasm is also not, by a strict definition, it's not an organelle, right? Because all the organelles are within the cytoplasm. So we say that it's a jelly-like material. You see here, jelly, therefore making, especially in animal cells, very flexible, therefore can assume different shapes depending on the type of cell. Even though there are some cells that do not have cell wall but still have quite a defined shape, like in red blood cells. Okay, all right. So the arrow here is pointing as to the where we have the cytoplasm. So the region just within the bounded by the cell membrane can be described as the cytoplasm. Okay. Alright, then we also have cytoskeleton, which, as you can see here, is made up of the we have materials like the microtubules, the filaments, then intermediate filament. So as its name suggests, this gives shape, okay, or framework to the cell, and more importantly, as you know, of skeletons tend to give it strength. It strengthens the cell. Okay, so these are protein fibers in cytoplasm. It gives shape, structure, and helps move organelles. Well, help is one word we are not supposed to use in biology, so we cancel it out. We take it out. I allow it to be there so that I can point it out to you. You know, in the WASI, it tends to be something that has led to some students actually not getting certain grades that they have to, because they use help most throughout. So rather than say help, you can just say give shape, structure, and move organelles. Just move organelles, that's all, or cause organelles to move. Okay. So examples are the microfilaments and microtubules. Okay. The intermediate filaments. So those are the three basic ones that we have, the three types that we have: microfilaments, microtubules, and then intermediate filament, which I said to give shape, structure, and move organelles. Alright, and we go to the centrioles and the most important organelle in cells. Okay, and as its name suggests, or its description suggests, right, cells control center. Okay, as a cell control center. So it tells you that it is a very crucial organelle. Just like if you take the whole human being and we are talking about the brain, take out the brain, they are nothing. In fact, even in the small part of the brain is affected. It affects, a certain part of the body as well, and mostly it is something that can limit the function of the human or the organism, for that matter. All right, so what does it have that makes it to actually be the control center? And that's the DNA or the chromosomes. All right, so we have chromosomes, and then the DNAs are in chromosomes. So condensed form, the DNAs are very long, thread-like, depending on the stage of the cell, whether it's going through some cell division or not. Thread-like or be a little thickened. So whatever it is, the DNA condensed form of it forms the chromosomes, right? So the two are almost the same, right? It's a condensed form of the DNA is the chromosomes. Okay, so what is so important about the DNA is that it contains the genes. The chromosomes, they contain the genes, and as you know, those are the inherited materials or molecules that forms part of our bodies. What genes basically do is to form proteins. Okay, and every part of us is made up of a protein. So they tend to lead to formation of gametes, lead to the formation of body parts and repair. And just like as we go on our day-to-day activities, we get cell part of our tissues and organs being damaged and all that. If you don't have a blueprint, something that we can look on and make exact copies of the torn or worn out part of the body, then it means that we are going to just die. So the genes have copies of every part of what you call the blueprints of the body, just like a mason will look on a plan to build a house, so that if there's a window, how do we design the window is already on the plan. So if you break your bone, right, the information to make a new bone or repair the bone is not in the hands of the doctor or the nurse. They just ensure that there is no infection, so and then the body heals itself by forming particular types of cells, whether bone cells, muscle cells, skin cells, hair cells, what have you, right? And these are all forms of mainly proteins, right? So this is why it's the control center. That's the chief reason, because of the presence of genes or DNA or chromosomes, right? Okay, so it's usually easiest organelle to see, usually one per cell. Okay, and in terms of the size, may be considered the largest at certain times of the cell development or cell division. Good. It has the nucleus, which makes ribosomes, and it's usually the dark spots within the nucleus. Okay, then we look at another organelle, that's centrioles. So you can see here in this animation, you see that these are centrioles that are moving towards particular part. So this is the nucleus right here. Then we have centrioles, right? A pair here, a pair there, moving to what we call the poles, right? And this occurs during cell division. At this point, the centrioles have reached the pole. They are the poles, and then we have these strings or threads. The diagram on the right. So you can see the chromosomes. They are attached to these thread-like materials we call spindle fibers, and then the chromosomes separate. We call this and this one chromosome, we call them sister chromatids. They separate at their a point of attachment, and then they are pulled as if being attracted by a magnet, to the pole. So usually have called the North Pole and South Pole. They were initially or originally aligned at the equator. Okay, at the equator. So a stage of cell division we call metaphase. So they align at middle. So they align at the middle or the equator, as we usually call it. And then at the next stage of cell division, which is anaphase, they begin to move to the poles. Okay, so you see the spindle labeled here, and then you have the centrioles there. Okay, right. So pull chromosomes apart during division, cell division. Cell division basically is the production of new cells, production of cells. So usually you have one cell forming two cells we call daughter cell. That's mitosis, one type of cell division. And then we have one cell forming four daughter cells, another one called meiosis, and that leads to formation of gametes. Cell division is ahead of us, so we'll look at it more when we get there. Yeah, so it's made up of microtubules, right? What did we say about microtubules? What are microtubules? Examples of which organ, right? So I want you to answer all these questions on our WhatsApp platform, okay, or in the comments and down there. So let's move on. They go to opposite sides of a cell when cell divides, right? As I've already said, only in animal cells. So we find them mainly in animal cells. Then we go to the ribosomes, right? Ribosomes are very important formation of proteins. So they are the sites of protein synthesis or making. They are found attached to rough air or floating free in the cytoplasm. So either they are floating or they are attached to endoplasmic reticulum, and that's what makes it to be rough, because once they are on the surface, it gives it a coarse, look or feel, if you like. So if you look at this one already. So in the diagram below, see the free ribosomes, then the ribosomes that are attached to the ER. Okay, right, and it says that they are the cell's protein factories. All right. So endoplasmic reticulum is sites where protein and lipids are made. They are attached to the nucleus, and if we have ribosomes on them, the surface, then we say that it's a rough endoplasmic reticulum, and those without it are smooth. I guess it makes sense, right? Okay, so for the rough endoplasm, because of the ribosomes, they make proteins, and then these ones without the ribosomes make lipids and get rid of toxins as well. Okay. Yeah, so you can see here ER and then the nucleus. Usually the ERs are heavily associated with the nucleus. So you see the nucleus here, right? And then the ER, okay, especially the rough ER, they are very close to the nucleus, right? And it is important because, see, during protein synthesis, another topic that is in either third year or so, or second year, I think third year, yeah. So during protein synthesis, a material is formed, or RNA, messenger RNA, that is transported outside the nucleus, formed in the nucleus and transported outside to come and attach to the ribosome, right? So this RNA, aptly called mRNA, that's messenger RNA, carries the information, the code, to form a particular protein. And I told you of the importance of what the genes and the as found on the DNA, right? We have all the blueprints to make proteins, part of us, or body part of the organism, right? So the mRNA carries this information, attaches itself to the ribosome on the ER, right? So as I've said, the rough ER is also associated with the proteins, in that when the proteins are formed, they are going to package it and then transfer it to the Golgi bodies, which will finally modify the proteins and then convert it into the suitable form that the body requires or the cell required at a particular time. If you are going to form enzymes, you transform that protein form into enzymes. If it is hormones that are required, it will convert it, and so on and so forth. So that's how they work. Now let's look at the Golgi apparatus, otherwise Golgi bodies. So the Golgi is the name of an Italian scientist who always writes with a capital letter starting as the gene. What does he do? So he stores, modifies, and packages proteins. Okay, so molecules transported to and from Golgi in vesicles. Right, so you see here the diagram. It has two ends. You have the trans phase and then the cis phase. So the cis phase, what does it do? Receiving, in quotes, the side of the Golgi apparatus. So it will receive molecules or substances usually from the ER. Okay, as I said earlier, and then it will go through the cystine, right, the various membranes, and then at the other end, it is now packaged, or it finishes the modification and then packages it, and then it will ship it. So at the trans end, that's where the ship is, which is the vesicle, like a boat, and then as you can see around it. So these are all finished products, if I should say, and transported either within the cell, or some can even go outside the cell. See this beautiful animation of the vesicles moving. So they are moving from ER to the Golgi, right? So the cis phase receives, and then the trans phase now finishes off at the other end, and then in the vesicle, like the bolt or a shape, carries it outside the cell, or even within the cell where it will be utilized. Okay, then we look at the vacuoles. The vacuoles store food, water, proteins, waste, and so on. And the large central water vacuole, usually in plant cells. So in plant cells, very large, almost as if it takes the position of the nucleus, as you see in the animal cell. So in plant cell, the nucleus is more to the cell, while the vacuole takes that central position. And it's very important, as you'll get to know, there are many small vacuoles in animal cells. So in plants, it has to be large enough, stores water, and which promotes the strengthening of the cell. When it takes in so much water, it stretches the cell wall, the cytoplasm in a vacuole, having so much water, then puts that pressure on the cell wall to strengthen it. Okay, and in young plants, seedlings, or coming in without any hardening or secondary tissues or so on, to strengthen it requires on these characteristics, what we call rigidity, to obtain that kind of strength to make it withstand strong winds, rains, and so on. Okay, so you see that when the winds come, they just bend. After that, they assume their previous shape. Okay, so the vacuole stores water and do all those things. Now we look at lysosomes. Lysosomes break down waste materials. Okay, and the lyso, or the lyso part of it means lysis, which means break down. Okay, some or somas, like a body, so like a breaking down body, soma. Okay, so this organelle is chiefly involved in breakdown of materials, breakdown waste materials. So you say the cells garbage disposal, isn't it? I guess it makes sense. Cells garbage disposal. And of course, for you to break down materials, just like you add some acid or some chemicals to certain materials to break it down, break certain waste down. Also use digestive enzymes to break down these materials. Just going back a little, and I'll pose a question here. The lysosome, in some cases, is also referred to as the suicide bag. Why is it a suicide bag? I want you to explain why it is a suicide bag, and you can post your answers later. Alright, let's move on to the mitochondria, which breaks down food to provide energy, referred to as the cell's powerhouse. But mind you, don't give it as a function, as a powerhouse, and it's an accolade of it based on its function, and we want that function, which is to provide energy. Well, that's where we have aerobic respiration occurring, and therefore it will use oxygen and glucose to make large amounts of ATP for us, which is adenosine triphosphate. The energy currency or the energy, form, of molecules. So the more ATP the cell is possessing, the more energy you have, the cell will have, and therefore the organism. Okay, so by way of structure, it has two membranes, the outer membrane, inner membrane. The inner membrane is folded into the cristae. Okay, so you see the many finger-like projections here. That's the cristae. One is crista, right? So in between the two membranes, you have the intermembrane space, okay, which is the outer membrane as well. Then inside you have the inner compartment here, and then the outer compartment. Okay, so you have the matrix within the inner membrane. Okay, then we look at the chloroplast. So the chloroplast and the mitochondria are the energy, transformers, if you like, of the cell, chiefly involved in materials that give us energy. With the chloroplast, so this is a drawing of a cell, typically a cell containing chlorophyll. See that is green, usually close to the cell wall, the chloroplast, and usually more at the periphery of the cell, or close to the cell wall or the cell membrane. So taking one, we have also, just like the mitochondria, two membranes, right? Inner, outer. Then we have the intermembrane space or the outer compartment. Then the inner compartment having the thylakoids, which are disc-like flat shaped, round structures packed together, something like a barrel. And when you have that, we call it the granule. That's granule for one. So many thylakoids packed together is granule. And then if you have more than one, like you can see so many of them packed in one chloroplast, and green containing chlorophyll, then we say is granule. Alright, and they are linked together by the lamella. So the structure that links them, that's the lamella. Okay, right. So we have this animation here, so glucose, the green, because at the end of the day, the chloroplast forms glucose, which is later changed into starch. So aside for food or glucose production, only in plant cells. But at this level, you now, having looked at other kingdoms, and then especially the kingdom Protista or Protista, found that the algae do have chloroplasts as well. They can be in different shapes. Some are star-shaped, called the stellate chloroplast. Some are like in some euglena, and then some chlamydomonas have the cap shape, and so on. So different shapes of the chloroplast, but then they all perform the same role, having chlorophyll, absorbing sunlight. Alright, then, so I've already said they have the chlorophyll, double membrane, and you have the equation, the popular equation that we write for that. So you have the sugar, which is glucose, which is C6H12O6, right? Then you can balance it. Okay, so we've looked at a number of the organelles. Now we are going to just do some comparison between animal cell, plant cell. So animal cells are usually smaller, plant cells are larger. No chloroplasts, plant cells have chloroplasts. No cell wall, plant cells have cell wall. No or small vacuoles in animal cells. Then plant cells have large central vacuole. As I said, nucleus is central, whilst is close to the cell wall in the plant cell as the nucleus. Okay, so this is a diagram just used to differentiate and then obtain the similarities between animal cells and then plant cells. So you can see that they have a lot in common, just few organelles that differentiate, or part of the cell that differentiate them. Okay, so so this is a recap of all that we have said. Right? Yes, for control, we have the nucleus, support, storage, and cleanup. We have cytoskeletons, we have the vacuoles, we have lysosomes, and to build proteins, we have the ribosomes, Golgi apparatus, energy transformations, we have chloroplasts, mitochondria, and then to put everything together, keep everything in shape. You can watch it as many times as you want. Remember to comment on anything, ask any question. I'll be more than willing to answer them. Right? Okay, so see you in the next one. Post one very soon.